A very big hello to all my wonderful PU2 as well as KCET champions. I hope all of you are doing absolutely great, and I welcome you today for today's Masterstroke series, where we are going to deal with the chapter Biotechnology Principles and Processes. So before I go ahead, I just want to tell you one very important thing. Every time in all of our lives, there might be one person who might feel like very rough and tough, but when you get to know them, you feel like, oh my god, this person was this sweet and kind, right? Similarly, this chapter is also like that. For so many of you, it feels like, oh my god, this is not biology, this is something else. Yes, it is biotechnology and it is very new for you, but trust me, if at all you make sure that you listen to this whole lecture, it will be super easy for you. It's all in your head that it's difficult. It's not difficult, it's easy, and you can grab those 7 marks very easily in your PU2 and also the 3 marks in the KCET for sure. Done? Can we go ahead? Let's go ahead. So the basis of this particular chapter is that we want to create DNA. Different type of DNA. What, ma'am? I didn't understand. Listen to me carefully. Suppose it's me. In my body, inside the cell, inside the nucleus, there is DNA, and inside the DNA there is one segment of DNA which is very important for formation of very important protein. This segment of DNA is responsible for coding a very important protein. Now, can I tell my body to produce it more? No, obviously I cannot. Okay, my body will not listen to me if at all I say like that. So basically, if at all I want the protein to be produced more, what I can do is that probably I can take the gene, I can take that gene and put it somewhere where it will create its own copies. Okay, so where am I going to put, my dear children? Usually, you put it inside a plasmid. Usually, you put it inside something that is known as a plasmid. So what you're going to do is that plasmid, it looks like this. This is also a circular form of DNA that you have. With the help of some enzymes, you will make a cut over here. Now you have an opening right over here. What do? You put the gene of interest, as in the gene that is going to code for the protein that I need, which is why I'll call this as gene of interest. Now what do? Basically, this particular plasmid is what? This particular plasmid is a recombinant plasmid now. Why do I call it recombinant? I didn't understand now. So listen, it was initially like this. I put a segment of my DNA over there, or my gene out there, right? And now it's a new combination that is being formed. Okay, it is a DNA only, but a new combination of DNA that is being formed. That is why I will call this as recombinant DNA. Okay, so until now only it's important? No. Basically, okay, I got the recombinant DNA and all, it's all good. It's there in the plasmid, it's all fine. And the plasmid, my dear children, as I said, is a circular form of DNA that is found in the case of prokaryotes. It is usually taken from the prokaryote itself. They have an origin of replication. Origin of replication means they have a site with the help of which they can replicate, they can create copies. But it's not just about replication. We all know to form the protein, the DNA which is there, it has to undergo transcription first. And then it has to undergo what? Translation. So that means, okay, replication part is sorted because I am putting it inside what? A plasmid. But now I want transcription and translation to happen. For that, I need machinery of the cell, which is why I would need to put it inside a cell, an actual cell, which is going to help me with all the enzymes needed for transcription and translation. So basically, this particular recombinant DNA, or the different type of plasmid that I have right now, I am going to put it inside a host now. What's a host? A host can be anything, probably a normal cell, which has all the machinery for doing transcription and translation. So I will be putting it inside that, and now the host will take over, do the same function, and over here I'll be getting my protein. So yes, it starts from here and it ends up over here when we actually get the products that we needed. But obviously there are more steps involved over here, and that is what we are going to discuss in this particular lecture. Okay, so we'll go one by one. First of all, I'll talk about all the materials that you require for this. Then later we'll talk about the process that is involved. Okay, done? Yes. And if at any point you feel that, ma'am, I am not understanding, pause the video, go back, listen to me and come back. Okay? And trust me, sit in this particular mind that this chapter is easy. I will try my best to understand it. That's all. And do not make your notes. You can make it after 30 minutes after pausing the video. Do not make it right now. Okay? Concentrate just here. Fine. Okay, so this is the whole story. I already spoke about it, but there is the first story which was there, as in the first recombinant DNA. How do you think it was made? The new type of DNA, how do you think this was being made? So this was made by two people out there. Their name was Stanley Cohen and Herbert Boyer, and it happened in the year 1972. It happened in the year 1972. Who all people? Stanley Cohen and Herbert Boyer. Now, the organism that they used over here was Salmonella typhimurium. Do you know this? Have you heard about this? Human health and diseases. Salmonella typhimurium causes what, Baba? It causes typhoid. Now, you know, it's a bacteria. It's a bacteria. So, bacteria has its own genetic material. This genetic material which is there, it is responsible for formation of proteins, everything which is needed for the cell to survive. Yes, yes. Apart from that, my dear children, you heard about the plasmid term. I use the term plasmid, right? What is the plasmid? So plasmid is something that is extra, but it is found in the bacterial cell, and it is extra chromosomal. It is extra chromosomal in nature, as in whatever is important is over here. They form the chromosome, they form the proteins and everything that is there. This is something that is extra chromosomal. Okay, it will have its own origin of replication. It will replicate by itself. It is not responsible for maintaining, or what do you call, for making sure that the cell is live or not. It's just there. Okay, it's just chilling out there. That is the plasmid DNA which is there. Okay, so it is extra chromosomal, it has its own origin of replication, and it is not involved in any production of proteins and stuff like that. Fine, fine. Okay, now listen to me. What if I tell you there is a gene, and this gene gives the superpower to any organism to become antibiotic resistant? That means, you know, to kill the bacteria, the Salmonella typhimurium, you take antibiotics. Now suppose I am saying, if at all the Salmonella typhimurium has this gene, it is going to be resistant. Even if you put antibiotic, it will still leave. Then what? Oh my God, that's crazy, right? Okay. And people want to experiment it. Okay, Stanley Cohen and Herbert Boyer, they really did want to experiment it, which is why they tried to construct a DNA like that, which has the antibiotic resistant gene as well. And they were checking whether it actually works or not. Okay, for that, what do they need? Listen to me carefully. First thing that they need would be the plasmid of Salmonella typhimurium itself. So you have the plasmid. What are you going to do with the plasmid? You are going to make the cut over here. Similarly, you are going to cut this particular gene. It also might be involved in some plasmid. You will make a cut over here with the same enzyme that is there. You call these set of enzymes as what? Restriction enzymes. So the first thing that you do is restriction enzymes cut the antibiotic resistant gene from the plasmid. So it was also a part of the plasmid. You cut this particular gene off, and now you have this. Similarly, my dear children, this thing which is there, this particular plasmid which is there, you call this as a vector. Why? Because it acts like a carrier. I told you, it acts like a carrier of the new DNA, and then we put this particular carrier inside what? The cell that is there, right? So I do not call it as a carrier. Rather, there is a particular name to it, and that is what? A vector. Vector is anything that is going to carry my gene of interest. So as of now, this guy is going to act like vector. So what did I do? I took a restriction enzyme. I made a cut over here, which is why, oh my God, just a second. I have a region like this. Now what do I do? In this particular region, I am going to put my gene of interest, right? And to make sure that it is very much attached, properly attached, you need the help of another enzyme that is known as DNA ligase. So what did you do? Very easy. First, you cut the restriction and you cut the antibiotic resistant gene and also the plasmid vector with the same restriction enzyme. Then what did you do? You basically inserted the gene of interest, and after that, with the help of DNA ligase, you joined it up properly. Now, so this plasmid is ready. What do you call this now? You will call this as recombinant DNA. This is a new combination of DNA that you have got, recombinant DNA. Okay, and it is going to act like what? The vector out there. Now basically, will you put this back inside the Salmonella typhimurium? Because if at all you do that, what is going to happen? You are going to basically provide the superpower of being resistant to an antibiotic. Oh my God, if at all it works, then it is a very big problem, because then we have to search for some other antibiotics to act against it, because it has got the superpower. So obviously we cannot take that risk of putting it back inside Salmonella typhimurium. Rather, we can try it on some other organism. So basically this particular vector is now put inside the next bacteria, which is known as E. coli. So inside the E. coli, I put this particular vector, my recombinant DNA. Now what is going to happen? Basically, at the end of the experiment, they found that E. coli, this particular E. coli, which had this recombinant DNA, was showing resistance. It was showing resistance to antibiotic. That means, oh my God, I actually created an antibiotic resistant DNA out there. And this was the first recombinant DNA, my dear children. Don't worry, they didn't put it inside the salmonella typhimurium. It was just separate, okay? It was done just to experiment. So I hope you have got an understanding. Do not forget it, okay? And yes, Stanley Cohen, Herbert Boyer, Salmonella typhimurium, and also E. coli was involved. The plasmid was taken from this guy itself, and then, my dear children, it was put, because we want to see that whether this plasmid is actually going to work, but we cannot put it back there, right? So at least some part of the Salmonella typhimurium had to be taken. That's why this particular plasmid was being taken. Got an understanding of it? Yes? Listen. So the gene is linked to plasmid DNA of Salmonella typhimurium using DNA ligase. Now creating recombinant DNA, a new circular DNA molecule is formed in vitro, that means in the lab. This is that. Now transferring to the host, the recombinant DNA is inserted into the E. coli. That is going to be known as what? The host cell. Now the plasmid replicates inside the E. coli, producing what? Multiple copies, and basically providing resistance as well. So that's all. 1972. Please do not forget the name. Okay. Now discovery of restriction enzymes. So I told you there were enzymes that were needed. Enzymes were also very much needed, right? So these enzymes that is there, before I go into that, please remember plasmid is extra chromosomal. I'll write it also here. It is extra chromosomal in nature, and remember it is circular in nature. It is circular in nature, extra chromosomal, circular, and it mostly acts like what? The vector. It has its origin of replication. Listen, vector can be anything. Plasmid can also be a vector. Some other vectors are also there. So it's not just plasmid. Remember, not all plasmids, not all vectors are plasmid. Not all vectors are plasmid. Always remember that. Okay, not all vectors are plasmid. There can be other things also which can act like vectors. Fine. So yes, that's all. So this is about just the plasmid. Extra chromosome is circular and it acts like a vector, and also it has origin of replication. Fine. Now, restriction enzyme that I was talking about. So the discovery of restriction enzymes in general happened between 1963 to 1968. So 1963, my dear children, 2 enzymes were discovered. They didn't know the name, they didn't know anything. Just 2 enzymes were discovered in E. coli, and it is a very strange story. In what? E. coli. So one which was there added methyl group to the DNA, and the other cut the DNA. So listen to me carefully. Over here, E. coli will have its own genetic material. Agreed? Now suppose a virus comes and attacks the E. coli. Okay, any virus which is there, suppose it is coming and attacking the E. coli, what does usually a virus do? Virus is going to transfer their genetic material inside the E. coli. They will be transferring their genetic material inside the host organism out there. Why? Basically, this genetic material which is there, it will slowly come and get incorporated over here. And now the virus is going to use the cell's machinery for its own production. That is how it usually works. Now E. coli got an understanding of this, and which is why they had their own defense mechanism. And 2 things came under their defense mechanism. First one is production of restriction endonucleases. What do they do? Endonucleases, my dear children. DNA is nucleic acid. DNA is made up of, DNA is what? Deoxyribonucleic acid, right? So this is a nucleic acid. If at all something is going inside the DNA and cutting it off within, this is known as an endonuclease out there. Okay, so basically this restriction endonuclease is going to cut the genetic material right over here of what? Of the virus. So ma'am, the genetic material is also here also. Why does it cut its own genetic material out there? There is a reason. So when I say it's a defense mechanism, you need to understand that yes, restriction endonuclease was the first one. The second one was one more enzyme. Okay, one more enzyme which was adding methyl group. which was adding methyl group. This particular enzyme went and added methyl group onto what? On the genetic material of the E. coli. What is the use of this methyl group? Whenever methyl group is there, whenever your genetic material is methylated, this particular restriction endonucleases could not act on them. It will just go act on them and cleave them. So it's like a signal that you do not have to cut me. You get it? So this was discovered in the year 1963. The name of the enzymes were not known. Just that two things were there. One added methyl group to the DNA. Why? Because it wanted to protect the DNA from its own defense mechanism, and the other cut the DNA, and you call them as restriction endonucleases. For the first time, with the name, the restriction endonuclease was discovered in, my dear children, HindII. So this Haemophilus influenzae, okay, Haemophilus influenzae was the organism where for the first time, my dear children, the restriction enzyme which was there, it was actually isolated and identified as well. Okay, so it was HindII. It was known as HindII. This is the name of the restriction enzyme. And always remember, restriction enzyme which is there, it has a specific region where it will go and make the cut. So this part, and you call that as restriction site. So this is the restriction enzyme that you have, and it makes a cut at a specific location. You call this as what? Restriction site. Okay, restriction site. And the restriction site of HindII, my dear children, has how many sequences? It has 6 base pairs. Okay, or basically 6 sequences are there in the restriction site. They have not mentioned what are the sequences, but please know there are 6 sequences over here. You call this as restriction site, or basically you also call this as recognition site. Okay, recognition site as well. So either way you can remember it. Now, it ensures precise DNA cutting at the same location every single time. So only at this particular location it is going to make the cut. Current knowledge, 900 plus restriction enzymes are discovered, isolated from 230 plus bacterial strains, and each recognizes different DNA sequences. So how many restriction enzymes are found? 900 plus. From how many bacteria? From 230 plus. And my dear children, each of them will be having different recognition site as well. So always remember, the discovery of restriction enzymes started from 1963 only. In E. coli, there was an understanding that there were 2 enzymes out there. But the actual one, the first one to be discovered properly with the name and everything was HindII. It was found in 1968. It was found in 1968. This year is important. And it cuts the DNA at 6 base pair sequence. Okay. And that is known as the restriction site or the recognition site. I hope that's clear. Yes. Now just give me a second. Okay, now my dear children, so if at all you heard me correctly, I told that HindII you get from Haemophilus influenzae. How did I come to that particular conclusion? How can you tell that, okay, this particular restriction enzyme came from this bacteria? There is an easy way. Okay, that is how it is being named, and we are going to see that as of now. So naming convention of the restriction enzyme. The most famous restriction enzyme that is there is EcoR1. Is what? Eco. I'll write it over here. EcoR1. Okay. Here E stands for the first letter of the genus name. Escherichia, you know, Escherichia coli is E. coli. Escherichia coli is what? E. coli. And from that you get EcoR1. So the first letter is taken from Escherichia, okay, that is E. Now the second letter which is there, second and the third letter which is there, CO. CO comes from what? The next 2 letters comes from the species name. This is, if it was coli, then basically the first 2 letters of coli are being taken over here. Now coming to R. R is what? The strain name. So there are different strains, okay. So it represented by an additional letter R, which comes by RY13. RY13 is the strain name of this particular bacteria. There are different strains, and depending upon it, you have different things. Now, what does the 1 stand for? EcoR1. The Roman numeral 1 stands for the order of isolation. So it was the first one to basically being discovered, which is why it is known as EcoR1. You got it? So indicates the order of isolation. First is the first enzyme from that particular strain which was being isolated. So I hope you got it. The first letter from the genus name, the second 2 letters from the the species name, and the this particular letter, that is R, which is there, you get it from the strain name, and the last number which is there, you get it from the, what it indicates, the order of discovery, or basically isolation, not discovery, basically isolation. Got it? Yes, that's all. Okay, now when you talk about restriction enzymes, you need to understand that you call them nucleases. Why? Because they cut the nucleic acid, right? And there are two types of nucleases. If at all you are a PU2 aspirant, you know that they usually ask the difference between exonuclease and endonuclease. So these are the two different types of nucleases that you have. Exonuclease, which is there, it is going to cut the nucleotides from the DNA ends. From the ends of the DNA, my dear children, you cut the nucleotides. That is the role of exonuclease. Now endonuclease is what? It cuts the DNA within DNA specific sequences within the DNA itself. So suppose this is very big long DNA out there, in the middle also it can come and make the cut. That is the role of what? Endonucleases. So endonucleases is something that we usually study more. Rather than exonucleases, we mostly study what? Endonucleases, because for the plasmid, for any DNA, we have to cut from the within itself, from middle itself. You get it? So exonucleases removes the nucleotides from the DNA ends. Endonucleases cut the DNA within the specific sequences. And there are some functions of restriction endonucleases which you should remember. How do they work? First of all, it inspects the DNA for specific recognition sequence. I told you the recognition sites or the restriction sites, it searches for that. All of them has a particular site, right? So similarly over here, suppose it is EcoR1, that is the enzyme that you are using. EcoR1 has a specific site, and what do you call the site as? You call the site as GAA-TTC site, and it is from 5' to 3' direction. If at all there is a site like this, it will go and make a cut out there. Make a cut where? It will make a cut between G and A. It makes a cut between G and A. That is how it works. So this is the restriction site or the recognition site, and it makes a cut between G and A. So first of all, it has to inspect the DNA for finding this particular site, and then bind and cut both the strands at specific points in the sugar phosphate backbone. Listen, if at all this strand is in this GAA-TTC sequence, you need to understand the other strand will also be in GAA-TTC sequence. So basically, it binds and cut both the strands. Okay, both the strands at specific points in the sugar phosphate backbone. You have to cut the backbone itself, and the backbone is made up of what? Sugar and phosphate. The sugar, which is your deoxyribose sugar, and remember the fifth carbon of it is attached to a phosphate. So basically, and then this particular phosphate will be attached to another carbon of the next nucleotide up there, which is why you call this as sugar phosphate backbone. Without this, the DNA is not going to be made. Okay, and if at all you want to break it, you would have to break the sugar phosphate backbone itself. Fine. Now recognize the palindromic sequence. So my dear children, over here, if at all you see the craziness over here is that both the site are being read in the same way, GAA-TTC, GAA-TTC, which is why you call the sequences as what? Palindromic sequences. From both the ends, it is read the same. If at all any Malayalis are watching this video, by the way, I'm also Malayali. Over here, if at all, I say if at all are way too much. Okay, Malayalam. Okay. If you read it like this, it is Malayalam. If you read it like this, then also it is Malayalam only. You get it? So basically, these sequences are known as palindromic sequences, which are read in the same way in both the ends. And always remember, in this particular case, in the case of EcoR1, the site is GA, Ga-T-C. Okay, take care. Ga, take care. So here 2 T are there and 2 A is there. Do not forget that. And it is 5' end over here, and it is 3' end over here. Do not forget that also. Okay, so we got an idea of how they work. Inspect the DNA, cut both the strands at what? Sugar phosphate backbone, and that's all. It recognizes the palindromic sequences as well. Fine. Now, when they make the cut, what is the use of this? That is very important. Restriction enzymes. Why do we say that? Basically, the gene of interest has to be cut with the same restriction enzyme, and also the vector which is there, it also has to be cut with the same restriction enzyme. There is a reason for that, and you have to listen to me carefully for getting to understand that. So over here, suppose, okay, this is the DNA that I want to enter. Okay, this is the DNA that I want to put it over inside the vector. What do I do? With the help of the restriction enzyme EcoR1, I cut it. EcoR1 will search for GAA-TTC site. It searched for it, and now it made a cut over here between G and A, and here also between G and A. So it is slightly changed. The cut will be right over here, okay, between G and A, and here also between G and A. What do you get? Here the G is gone, okay, and here the G will be left. This particular strand is not going to be there. When you make a cut like this, what is going to happen is that this L-shaped thing which is there, it will gonna go. Rest of it is attached to the DNA, it will stay out there. You get it? So basically, one strand is unpaired. The hydrogen bonds are waiting. Oh my God, where is my other pair out there? You get it? So this is going to form a sticky end. It is sticky. Why? Because it has not formed any base pairs yet. There is no pairing that is happening, which is why you call them sticky ends. Now what if I tell you I can give you the exact sequence? Yes, which is why, and how will you get the exact sequence? If at all you cut with the same restriction enzyme. Over here the vector DNA, that is I'm talking about the plasmid. Okay, the plasmid where I'm going to make this particular foreign DNA put inside the plasmid, right? So this particular plasmid, if at all I cut it with the enzyme that is EcoR1 itself, it will make a cut over here between GNA, here also GNA. What is going to happen? This particular thing is gonna, this particular thing which is there, it is gonna go because rest of it is attached to the DNA. You get it? So this is going to go, this goes by. Now what do you have? Here you have sticky ends, here also you have sticky ends. Both the sticky ends, when they join, my dear children, there is complementary base pairs, which is why they are gonna make quick hydrogen bonds. They will be making quick hydrogen bonds, and tada, you got the recombinant DNA. You get it? So this is, suppose, what do you call, a plasmid out there. This is suppose a plasmid out there. In one end of the plasmid you made it cut with the GATTC. Similarly in the other end also you do that, which is why the foreign DNA which is there, it will also have something like this. Both the ends will be having sticky ends. Here also both the ends will be having sticky ends, and the sticky ends will be joining. Here only one end is being shown. Only this side is being shown. You get it? Similarly, that will be happening on the other side as well. So sticky ends formation is super important. So first of all, it cut away from the center, creating single-stranded overhangs. Okay, so what is going to happen? Whenever restriction enzymes are going to cut, please remember they will be forming what? Single-stranded overhangs. So there is an overhang that is being made, and it is single-stranded as well. Sticky ends helps in hydrogen bonding with the complementary sequences out there. And what is the use in genetic engineering? It creates recombinant DNA from different sources. DNA fragments cut by the same restriction enzymes can be joined using DNA ligase. So yes, sticky ends are enough to join them, but still there might be some places where they are left behind, and here comes the role of the very important enzyme that is known as DNA ligase. So DNA ligase, you can just remember it as the Feviquick. Okay, the Feviquick joins everything, right? Similarly over here, DNA ligase is just like that particular Feviquick, which is going to join things very smoothly. If at all there is a little nook and crack out there, it will be sorting it out. Got it? Did you understand it? I hope it's clear. Yes, so that is DNA ligase. About DNA ligase we studied over here also. Where did you study? Over here. Yeah, remember DNA ligase was used to chipcow this. So as of now, when I teach you more and more, what is going to happen is that basically I am pretty sure you are able to understand things in a better way, right? A little better way, 5% better, 5% better. So yes, after some time you will feel all the more confident. And also I do not want you to go through the entire chapter all at once. You can just pause the video. As I said, when I ask you to pause, you can pause the video, go back and just go through. Okay, done, fine. Now okay, so restriction enzyme part is clear, but I have a request. As I told you, EcoR1, you know, you get it from Escherichia coli. Similarly, if at all you are given any enzyme out there and you are asked from which particular bacteria or which particular organism you get, check, okay, whatever the term is given over here in the enzyme's name. Okay, hint to, as I told you, H stands for Haemophilus. I and N stands for influenzae, Haemophilus influenzae. So I and N from influenzae. So basically I got to understand that this particular enzyme I am getting from Haemophilus influenzae. So similarly, there can be any enzyme given to you. This is for KCET I am talking about, and you would have to find the organism from which you got it. So just use your basic understanding out there. Okay, that's all. And do not forget the site of E. coli. Now, I told you that it's not just plasmid that is going to act like a vector. It can be anything out there, right? And the best example for that is seen in the case of animals. You know, in the case of animals, if at all you want to insert a gene, okay, if at all you want to insert a gene inside the host cell, plasmids obviously cannot work because in us, the plasmid is generally not there, right? So what do we need? We need the help of some retroviruses. You know, this might shock you, but listen to me very carefully. HIV, how does it work? HIV has its own genetic material. It comes and puts its genetic material inside our cell, converts into DNA, and basically this DNA is incorporated into our nucleus, our DNA out there, and it makes its own copies out there. Right? So this is the exact mechanism that we are going to use. But over here, the retrovirus is not going to transfer its component. It is rather going to transfer our gene of interest. Whatever gene of interest that we want to transfer, it will transfer that. So since it is transferring that, I will call this as what? A vector. So retroviruses is the vector that we have for animals. Always remember. That is the only thing that you have to remember in the case of animals. If at all you have a vector, and that is going to be one and only retroviruses. Think about HIV. It naturally transforms normal cells into cancer cells. It has that ability. How does it turn a normal cell to cancer cell? Again, it has some genes. When it transfers the gene inside a normal cell, our normal cell becomes cancerous. So which is why I am telling you, it can transfer the gene of our interest also, right? If at all we do some modification out there, yes, that exact mechanism is being used over here. So what do? We use modified retroviruses, not normal retroviruses. We use modified retroviruses and to deliver desirable genes into the animal cells. And also remember, they are disarmed. By disarmed, I mean that they do not have any things like that. Okay, so no sword, nothing like that which can infect us. They are totally disarmed. Any genetic material which can cause us trouble, that is being removed, and it's a modified thing that we have. Once the gene is inserted into the vector, it is transferred into the bacterial cell. Okay, fine. So this is about animals. Remember, for animals, if at all you are being asked about a vector, it is going to be retroviruses. If at all you go write something else and come, my ghost is not going to forget you. Okay, now listen to me. In the case of plants, how do we do the transformation? Okay, or basically how do we transfer things out there? It's very easy, very, very easy. Listen to me very carefully, okay? So for that, again, we would need a bacteria's help. We would need the help of a bacteria. So this particular bacteria is very famous for causing a trouble. The name of this bacteria is Agrobacterium tumefaciens. What do you call this? Agrobacterium tumefaciens. It has its own genetic material as a normal bacterial cell out there, and as I said, it has a plasmid. It has a plasmid. This plasmid has a specific name. You call this as Ti plasmid. Okay, ma'am, why a specific name? Do you know what's the meaning of TI? TI stands for tumor inducing. It basically is inducing tumor in the case of organisms. So this is TI plasmid that we have. Agreed. Why do you say that it is tumor inducing? Because there is a gene right over here which is known as tDNA. And this particular part, my dear children, whenever a bacteria is going to infect a plant, the plasmid gets inside the plant, and also so does the tDNA. This tDNA is going to cause what? A huge lot of trouble out there. It causes tumor in the case of plant. It causes tumor in the case of plant. Why? Because this particular part is being transferred. What do you call the tDNA as? tDNA stands for transfer DNA. So the DNA that is actually getting transferred and causing trouble is this particular guy. So this plasmid is Ti plasmid. It has a DNA that is tDNA, which basically is the root cause of all the problems in the case of plant. It usually affects the plant. This bacteria affects the plant, transfers the tDNA and causes tumor. But how do we utilize it for our purpose? If at all, suppose I want to give the plant a pest resistant gene, a gene that will give it a superpower to be resistant to some pest out there. How will I do that? Basically, I know how it works. I definitely know how it works. So what can be done? Basically, why not just remove this particular tDNA part? Okay, why not just cut off this tDNA part and insert the gene of interest? So this is my gene of interest. I will be putting that in. It is still going to affect the organism. It thinks that I have the tDNA, but it does not have the tDNA. It actually has the gene of interest. So gene of interest is now going to be transferred. So isn't it acting like a vector? Obviously yes. Obviously yes. So my dear children, vector in the case of plants. In the case of animals, you know it is retrovirus. In the case of plants, it is going to be what? Agrobacterium tumefaciens. Always remember, it transfers what? tDNA or transfer DNA into the plant cells, causes what? Tumors. Now modified Ti plasmid. This is a normal Ti plasmid. tumor-inducing plasmid. If at all I modify it, and I can be using this as what? A cloning vector, non-pathogenic, the one that does not cause any problem. Okay, to insert what? The desired genes into the plant. Now this desired gene can be anything, pest resistant, this and that, whatever it is there. Okay, so please remember these two vectors which are there that you need. We read about restriction enzymes, that is clear. This vector is also clear. And now, once the gene is inserted into the vector, it is transferred into bacterial, plant and animal host wherever it multiplies. So here I spoke about plant and viruses. Apart from that, we also spoke about bacteria where, as I told, the plasmid of Salmonella typhi was put inside the plasmid of this guy. So it's the plasmid that usually acts as a vector out there. Okay, now there are some vectorless methods also. Okay, there are some methods where you do not need a vector to transfer things, and that is known as vectorless method. How does this thing work? It is different in different type of organisms. Let's talk about bacteria first. First of all, my dear children, you need to understand a term that is known as competent. I say that vectorless methods are there, which makes the cell competent. By that I mean these cells have the ability to take up the DNA from the surrounding. That is what I mean by the term competent. So when I say that I want to make a bacterial cell competent, I am meaning that I want to make that cell able to take up the DNA from outside. That is the meaning of competent. Okay, got it? Okay. Now listen to me. What do we do is that, suppose this is the bacterial cell that we have. And here we are going to have our DNA, the gene of interest that is there. It is right inside the medium. Now, first you are going to treat it with divalent cations, as in the valency will be di2. Okay, divalent cations, for example, calcium. Okay, so what do? You are going to treat it with calcium chloride. If at all you talk about the medium, it will be calcium chloride, but the main guy is this guy. Why? Because it is a divalent cation. Now what is the use of a divalent cation over here? It is going to neutralize the negative charge on the DNA. You know your DNA is negatively charged. Okay, your DNA is negatively charged. Why? Because it has phosphate group in it, right? So what do? We neutralize the negative charge out there. What happens if at all you neutralize the negative charge out there? Basically, it will be able to get inside the cell very easily. Okay, because our cell membrane also have some charges, you know, it will repel. There are so many things that can happen, but if at all everything is neutralized, obviously this can pass very easily inside. So neutralizes the negative charge. Apart from that, there is one more function that this does, and that is, so divalent cations neutralize, that is definitely there. Apart from that, they increase the permeability. They increase what? Permeability. Permeability means ability to take up the things. There will be more spaces in here such that the DNA can be taken up very easily. So first blocker was the charges, but now it is neutralized, and also it is increasing the permeability. But obviously even after doing this, the bacterial cells will not quickly take up the DNA out there. For that you need something else also to be done. So first of all, what you do is that you incubate the cells with DNA on ice. First you put this on ice. Then, my dear children, you are going to transfer it into a hot medium. So briefly, you're going to heat it at 42 degrees Celsius. That is more than our normal temperature out there, 42 degrees Celsius, and this is known as heat shock. So from ice, basically you are now heating it up at 42 degrees Celsius, my dear children. So it's like a shock. And when this happens from cold to heat, my dear children, what is going to happen is that there will be lot of force being created, which is why it can quickly. So basically, when ice was there, it was all normal. When you put it in the heat, the temperature changes. Basically, all the pores which are there, they will be opening up, and which is why the DNA can quickly get inside. And now what do? Again, after ice, you put heat shock. You put it in heat shock. Then again, you put it in the ice. So everything is sealed back again. Everything is going to be sealed back, and by this time the DNA is going to be inside. So this forces the bacteria to take up the recombinant DNA. This is one such method. This is one such method that we have. Remember, DNA is hydrophilic. Hydrophilic, it is water loving, whereas our cell membrane is hydrophobic in nature. So it cannot pass to the cell membrane very easily, which is why first thing that we have to do is treat it with divalent cations. It is going to neutralize the charge, and which is why here hydrophilic and hydrophobic would not play play any role, because after neutralizing it is very easy for the cell to take up the particular DNA out there. And not just neutralization, divalent cations which are there are also responsible for increasing permeability. But still, if at all you want to make the DNA go inside the cell, you need to give some treatment from ice to heat shock and then back to ice. Got it? This is the way that you make the bacterial cell competent. You make it able to take up the DNA from outside. Cool. Here, do you use any vectors? No, there is no vector out there. That is why it is a vectorless method. Now, when you talk about the plant cell, so I hope I can remove this if at all it's okay, or I'll just go there. Okay, listen. Now, listen to me carefully. When you talk about a plant cell has a cell membrane and then it also has a cell wall. Agreed? This is what? This is a plant cell. Now, when you talk about an animal cell, it just has a cell membrane and that's all. And that's all. So, it is pretty much easy out there. So what are you going to do is that basically there is a method that is known as micro-injection. With the help of micro-injection, basically I can just go inside and target the nucleus right there. I can use a pipette with the help of which I can transfer my gene of interest inside the nucleus directly in the animal cell. Why? Because there is no cell wall, there is no one to hinder. I can just do it. This is micro-injection. This is done in the case of animal cell. Directly injects recombinant DNA, the new type of DNA that we have, the gene of interest that we have, into the nucleus of an animal cell. But can you do it with the plant cell? No. Why? Because the plant cell has a cell wall, will not allow this thing to happen at all. So for that, what do you need? You need a gun. Oh my god, actually yes. What do you call this gun as? You call this as gene gun. And you have other name for the same thing. You also call this as biolistic, and this is what children usually forget. So biolistic and gene gun is the same thing itself. But what are you going to do? You are going to put in some gold and tungsten particles which are wrapped up with our DNA, and my dear children, you put it with high velocity inside the plant cell. So high velocity gold and tungsten, please do not forget, particles coated with the DNA are shot into the plant cell because it has a cell wall. It cannot undergo micro-injection method. Got it? Got it? That's the only thing. Now disarmed pathogen vectors, okay. So that is not needed over here. So these are the things that you need to remember. So this is micro-injection is for the animal cell. Is there any vector over here? No. That is why it is a vectorless method. Biolistic or gene gun is for plant cell. Is there any vector over here? No. Again, that is why it is known as vectorless methods. So we saw the vectors which are being usually used apart from plasmid in the case of animals as well as plants. We also saw vectorless methods. Restriction enzymes are done. And now, my dear children, I want to talk about one very important thing, and that is KC30. So I hope at least some of you might have heard about KC30. It is KCET 30-day crash course that we have, where there will be complete syllabus coverage, that is PU1 as well as PU2. There will be chapter-wise KCET questions in the test format. And my dear children, by that I mean all the previous year questions are going to be covered. There will be live doubt clarification classes. Okay, there will be live doubt clarification classes, and also additionally there will be 10 full-length KCET mock test. Okay, what do you have? 10 full-length test KCET mock test is there. This is very, very important. So your mock tests are being sorted over here. Your previous year question revisions are also being sorted over here. Your complete revision is also being sorted over here. Listen, this is not the correct time to prepare for KCET alone. This is a time for preparing for giving more priority for second PUC. You know that. You have your exams coming up. So I'm not asking you to go take it up and start studying from now. No, but you can take it up, this course, you can keep it ready. Why? Because you will need it just after your exam ends, because you just have a 30 days gap. That is exactly why this batch is known as KC30. So these 30 days which are there are going to be efficiently utilized. The start date for the program is 21st of March 2026. Okay, so just right after the exams end, my dear children, soon it will be beginning up. And over here the pricing for this is rupees 2499. Now remember, this is an offer price. It is going to end on 25th of Feb. That is exactly why I'm promoting it right now. So 25th will be soon coming up. Do not forget to take the KC30 course, and yes. And obviously you can do your own research about how the courses are from Diksha Karnataka team and things like that. It is definitely going to help you all. Yes, yes. Now, You can officially take a break if at all you want and go through whatever you have studied until now, because there are high chances that we might forget things, right? That is exactly why I'm telling you. But if you think that you're all good, okay, then we can go ahead also. Okay, so next thing, the features of a vector. We spoke about all the things. Now vectors features are also there, which are super important. First thing that a vector should be having is that origin of replication. Here I am specifically talking about a plasmid actually. Okay, in actual sense I'm talking about a plasmid. So origin of replication should definitely be there. What is this point? This is a starting point where the DNA polymerase will come and attach and do the DNA replication. Whenever you want to make the multiple copies of things, obviously you need origin of replication. So it controls what, Baba? It controls the copy number of the linked DNA also. So when I say this is a plasmid and this plasmid has my gene of interest right over here, and I am saying that this plasmid has an origin of replication which allows this much copy number, so that means 12 copies of the same gene will be made, right? So it controls the copy number of the linked DNA. That is what I mean. Now vectors with high copy number ORI help recover more copies of the target DNA. So basically, if at all a vector has it has an ORI which supports high copy number, obviously it is going to be really good. So first feature that should be there is what? Origin of replication, and it is the one that controls the copy number. Do not forget it. Second thing, selectable marker. Now here I am going to talk about two terms which could have been difficult for you but will not be difficult for you from this point onwards. One is transformants, another one is recombinants. Okay, listen to me carefully. Suppose there is okay, so here I have to talk about PBr322. There is a plasmid like this. This plasmid has origin of replication right over here. Okay, it has a site for ROP. ROP is what? A very important protein that you need for replication. So this site forms that particular protein. ORI, you know, its origin of replication. This is where the replication process is going to start. Apart from that, there are two genes out there. This gene provides resistance to an antibiotic which is known as ampicillin. So ampicillin is an antibiotic and it provides resistance to that. If this gene is there, this organism that has this particular plasmid will be resistant to ampicillin. Similarly, the other gene which is there, it provides resistance to tetracycline. This is another antibiotic which is there. Normally, a normal plasmid does not have all these genes. Okay, normally a normal plasmid does not have all these genes, which is why a normal cell will not be resistant to what? Ampicillin or tetracycline. And also, my dear children, if at all you want to check one mock test, if at all you want to just give one mock test out there, in the description section of this particular video, you will be able to see a link that is free KCET mock test PDF. If you click on that, you would have to give your mobile number and you will get the access to one particular PDF. You can try for that as well. Okay, so now let's go ahead. Suppose I am telling a cell has PBr322. I have put PBr322 plasmid over here into one particular cell. What do I mean by that? I mean that now it is going to be ampicillin resistant and also tetracycline resistant, right? Since it has taken up something from outside, it was not its own, it took it from outside. Initially it was not like this, which is why I'll say that it has underwent a process that is known as transformation. It has underwent a process that is known as transformation, and I'll call this as a transformant cell. Transformant cell. Why? Because it has taken up something new from outside. Okay, I'm not saying that it has a new DNA also attached to it. No, it has just taken up something new. Cool. This is what? A transformant. Fine. Now listen to me. What is the use of these genes out there? They are going to act like selectable markers. How do they act like selectable markers? The question is very simple. You need to listen to me carefully to give the answer for that. This cell was initially like this, where a normal plasmid was there, no resistance was there. So no resistance to ampicillin or tetracycline. No resistance to ampicillin or tetracycline. Here, this is what? A transformant cell. Okay, this is the same thing that we had. Now you have an additional thing right there. This is a new thing that you have got. This is now known as what? A transformant cell. Now how do I understand whether which one is transformed and which one is not? Which one is a normal cell out there? I just grow them in a medium that has ampicillin or tetracycline. If at all they are able to live in that medium, it means what? It means that they have underwent transformation. If they are dying, it means that there is no new plasmid out there, there is no transformation that has taken up. So don't you think that because of the presence of these two genes, you're able to select them? Why? Because these two genes provide resistance, and that is how you're able to select them. Which is why I'll say that these genes act like what? The selectable markers. It helps identify the transformants, the cell with the inserted DNA, the new DNA out there, and eliminates a non-transformant cell without the inserted DNA. Antibiotic resistant gene, ampicillin, tetracycline, chloramphenicol, kanamycin used as markers. They are used as what? Markers. Listen to me carefully. This is very, very important. A normal cell is there. It does not have all these genes. If at all it has taken up a new plasmid out there, then only it will be showing up this feature of being resistant, which is why I'll call the genes which are there as selectable markers. It helps us in selecting the transformants. Okay, the one that has taken up new things out there from outside. Got it? Yes, got it. Done. Selectable markers should be an ideal feature of plasmid. That is very, very important thing. It should be there in a plasmid. Then we say that this plasmid is really, really good. Now listen to me carefully. So this is PBr 322. Now, over here, I want your attention. Until now, we have not spoken about recombinant cell. We spoke about transformant cell, that the one that has taken up the new DNA. We didn't speak about recombinant cell. So, the next feature of a vector is that it should be having cloning sites. Cloning sites as in sites where I can insert my gene of interest. Okay, and it should be having multiple of them. So I try to mean that. Suppose over here I have a site at which BAMH1 enzyme can act. Basically, they can come and cut over here and they can put the gene of interest. They can help us in providing a space where we can put the gene of interest. This is BAMH1 recognition site. Okay. Similarly, over here I have another enzyme recognition site, that is SAL1s. Again, I can use the enzyme SAL1 also for doing this function. Okay, so I have multiple options out there, because each of them, it's not like BAMH1 has sites anywhere else. No. If at all it has multiple sites, what is going to happen? It is going to make a cut over here. Suppose it makes a cut over here, everything will be gone. So always remember, one enzyme will have only one region, one recognized site. You can have multiple enzymes out there. Okay, one enzyme, one cloning site. Always remember, one enzyme, one cloning site, multiple enzymes out there, and each one of them having their own cloning site. That means at the end you get multiple cloning sites as well. That is what you mean. Okay, it does not mean one enzyme, two cloning sites. That is wrong. So this is BAMH1 site and this is SAL1 site. Okay. This is a gene that you have for tetracycline. In that only you have the sites for these two enzymes. Similarly, over here you have PVU1 and you have PST1. Okay, BAMH1, SAL1, PVU1, PST1. These two enzyme sites are right over here. In ROP site, again you have one thing, enzyme site, and that is known as PVU2. So PVU1 is here, PVU2 is right over here. Apart from that, you have three enzymes over here. One is EcoR1, the most famous enzyme. The second one that you have is CLA1. CLA1. Then you have one more which is shown like this. This is HIND3. The HIND2 was the first discovered one. This is HIND3. So listen, BAMH1, SAL1, PVU1, PST1, two on both the sides. Here you just have one, and that is PVU2. Here you have three. So 1, 2 and 3. EcoR1, CLA1 and HIND3. You have to remember all their names. It is important. It is important. And you will remember it. It's not like a humongous task. You will remember if at all you make by yourself. Now, there is a problem. The problem is that, suppose I am using BAMH1 for basically providing a space over here and putting my gene of interest. You need to understand that when it does that, there will be a new gene being inserted over here. New gene. So do you think it will be tetracycline resistant anymore? Do you think it will be tetracycline resistant anymore? No. Why? Because you have changed. You have put a new gene over here. It is not going to be tetracycline resistant anymore, which is why I'll say that it can lead to insertional inactivation. It can lead to insertional inactivation. When I insert a gene of interest, the older one which is there, it gets inactivated. It gets inactivated. So my dear children, remember that also. So over here, whenever I'm using BAM-H1 or SAL1, or if I use PV1 and PST1, either way, it is going to mess up with the resistant gene out there. It is not going to show their original function. Now this exact thing is actually utilized for our purpose. For our purpose, you might think, how ma'am? So basically, listen to me. First we'll deal with this and then we'll talk about that. Vector should have a few or single recognition sites for restriction enzymes. Okay. Too many recognition sites, multiple fragments, complicate cloning. So for one restriction enzyme, only one site. You can have multiple restriction enzyme, multiple and there 111 cloning site as well. That is also okay. That is, totally fine. The way I just told you before. This chapter is confusing actually. Okay. Fine. Let's focus. So over here, with the information that we have now about the features of the vector, I am going to make the best use of it and I'm going to tell you how does things work. So listen to me very carefully, okay? There are high chances that I might get confused while teaching you, but I try my best not to be. So over here, suppose I have one cell like this and one cell like this and one more cell like this. Okay, so basically over here this is a non-transformant. That means this one does not have my PBr 322. That is for sure, and I'll tell you how are we sure for that. Okay, here I have it. Here I have ampicillin resistant and it is tetracycline resistant, both. That means a normal PBr 322. Fine. Just before doing this, you need to understand that we did one more thing. With the help of BAM-H1, we've made a cut over here and we put our gene of interest over here. We put what? Our gene of interest over here. So do you think it is tetracycline resistant anymore? No, that is gone. So this one, it is not tetracycline resistant anymore. And this one, since it has my gene of interest, I am going to call this particular plasmid as what? My recombinant plasmid, because it has a new combination which was usually not there. This is a recombinant DNA or also a recombinant plasmid out there. Always remember. And where am I putting it? Suppose I am putting it inside this. Suppose not, I am putting it, it took it. Okay, it took it. And over here I have that. So this is still ampicillin resistant because you didn't cut it with PVU1 and PST1, you cut it with just BAM-H1. And over here you do not have tetracycline resistant, instead you just have the gene of interest. Okay. Basically, how do you understand which one has taken up the new plasmid out there and which one has not? I'm just telling you which one has taken up PBr 322. I do not know whether it has taken up recombinant one or the normal one. I just want to know which one has taken up the new plasmid and which one has not. So what do we need to do that? Need to do is that we just need to put it inside the ampicillin medium. When I put inside the ampicillin medium, what is going to happen? The ones that have taken up any form of new plasmid, PBr322 from outside, whether it is recombinant form or whether it is normal form, basically they are going to survive. Why? Because you are putting in ampicillin medium and you have the ampicillin resistant gene in PBr322, which is why they are going to survive, and which is exactly why I'll say that these two cells which are there, they are what? They are transformants. And I told you, I already told you that this is not transformant. Why? Because, my dear children, this cell is not gonna survive. This cell is not gonna survive. Why? Because it has not taken up any plasmid from outside. It has not taken up any plasmid from outside. That means it is what? Non-transformant. So transformants means anyone who has taken the external or the new plasmid from outside. It has nothing to do with recombinant DNA or a normal DNA out there. If at all you have taken something from outside, you are basically said that you have underwent transformation and you are now transformants. So these two cells are transformants. I got to understand it when I grew these cells, or basically when I put these cells in a ampicillin resistant medium. So these two cells are being spotted. So I'll put the numbering of the cell, numbering 1, and this is numbering 2, and the third cell. So third cell is out of the league because we got to understand what is a transformant. How were we able to do that? With the help of selectable markers, especially ampicillin resistant gene out there, right? Now we need to understand which one among them is a recombinant. Which one among them is a recombinant? That means it has which one has my gene of interest. How do you find that? So for that, you need to do one more plating. That is why you say you need double plating in this particular case. One more plating, and this time in which thing are you going to grow it? You are going to grow it in tetracycline medium. Tetracycline medium. Now listen to me carefully. Over here I have the first cell, and over here I have the second cell. This is the first cell, this is the second cell. The first cell, as you know, it was a normal PBr322, which is why it will have ampicillin resistant gene and also tetracycline. Here there will be just ampicillin, and there on the other side it will be what? My gene of interest. Why? Because this is the recombinant. This is the recombinant, and this is actually the non-recombinant, the one that does not have my new DNA out there. So the one that has the gene of interest, you call them as recombinant, and the one that does not have the gene of interest, it is transformant. Okay, it has taken up the PBr322 but did not take up the PBr322 which had my gene of interest. So which is why I'll call this as what? Non-recombinant. But how will I know that? When I grow both of them in tetracycline medium, my dear children, the recombinant ones will not be able to grow. Why? Because now it is not tetracycline resistant anymore. I have put my gene of interest there, which is why it is not tetracycline resistant anymore. Whereas on the other hand, these cells are gonna grow. The non-recombinants which are there, they will be growing. Why? Because it has the tetracycline resistant gene. There is no gene of interest. So which one will I choose now? I will be choosing the one which does not grow. I will be using this for my further experiment. And over here, this is more complex. Why? Because you need two medium. Double plating is required. I hope this is clear. Transformants and recombinants are different. Recombinants are the one that has the gene of interest. Transformants means any cell that has basically taken up a newer plasmid or newer thing out there. Got it? So both of them are transformants, but only one of them is a recombinant. Got it? I hope with this you have understood this very important thing. If yes, please let me know. And yeah, fine. So this is a HOTS question also that came in, and it was there in your preparatory also, and I believe now it is strictly sorted. So PBr322 is one such plasmid. You need to remember that. Apart from that, insertional inactivation is seen over here. In one more place also you see insertional inactivation, and that is found in LacZ gene. So I'll just talk about that and then I'll be solving that particular question. So over here there is one more setup, one more type of plasmid where you have the LacZ gene. LacZ gene, my dear children, you know ZYA in lac-operon, what does it do? Z1, it is coding for beta-galactosidase. Beta-galactosidase. This is the normal function that is there. And usually, if at all you are adding some chromogenic substance, as in some coloring substance, chromogenic substance over here, what is going to happen? This beta-carotesidase is going to bring out a change in this chromogenic substance, giving a blue color colony, blue coloration. Giving what? Blue coloration. This is the normal trend that you see. Cool. Now, so overcomes the need for dual plating. Wait. So this is a normal thing that we usually see. What are we going to do is that over here, instead of the LacZ gene, we are going to put our gene of interest. So what will happen if we do that? If we put our gene of interest at the same location, just give me a quick second, okay? Just give me a quick second. Okay, so my dear children, sorry, someone knocked the door. Okay, so right over here, what I'm going to do is that instead of LacZ gene, I'm going to put my gene of interest. If I put my gene of interest, do you think beta-carotesidase will be there? No, Baba, it won't be produced. If beta-carotesidase is not being produced, do you think blue color colonies are going to be, or blue coloration is going to happen? No. Even if you put chromogenic substances, you will not find blue coloration. So what I'm going to do is that, what I am going to do is that, I'm going to replace the PBr322 thing one with this one. Why? Because in that you needed dual plating. But over here, in one single plate itself, you get an understanding which one is recombinant and which one is not. So it's very simple in comparison to that. Okay. So what is the use of it is that, basically, if in a plate I put both the cells, okay, the cell that is having the plasmid with my gene of interest over here, and the cell with the plasmid that does not have my gene of interest. I'll show it by yellow. So this one is suppose first, this one is the second one. So this I put it in this cell, and this one I put it in this cell. Okay. So this one and this one, both of them are transformants. Why? Because they have taken up the new plasmids for sure. But which one is recombinant out of them? Only first one, only the first one. And now I am going to put the chromogenic substance in this particular medium. I would be able to see that towards one side there will not be blue color colonies that will be seen. Yes, definitely the cells are going to divide, but over here you are not going to see blue color colonies. Instead it will be plain white in color. The white color colonies will be seen. Why? Because here you do not have your what you call beta-carotesidase to give the blue coloration. You instead have what? Your gene of interest. On the other side, my dear children, if at all you see this side, the cells will be dividing, and over here you will see blue color colonies. Why? Because you still have the beta-carotesidase enzyme to do this. So basically in one single plate only you are able to understand this. Why? Because there is two different coloration being formed, blue and white. The blue one is the one that is non-recombinants, and the white ones are the ones which are recombinants. Always remember that. Okay, so here foreign DNA is inserted into the beta-carotesidase gene inactivated, and now chromogenic substance added. The non-recombinants, no insert, produces blue colonies. I told you here you get blue, and the recombinants which are there you will get white ones. So which one is better in both of them, PBr322 or LacZ gene, or the plasmid with LacZ gene? Obviously the plasmid with LacZ gene. Until here, if at all you have understood, please put down the comment section that, ma'am, it's all clear. I really want to know that. And now there's a question that I want to ask you. ROP gene, which codes for the proteins involved in the replication of the plasmid PBr322 in E. coli, is located at the restriction site of what? So this is 2021 question of KCET. So they are asking you in the ROP site, which is the restriction, in the ROP gene which is there, which is the restriction enzyme site which is there. It is HIND3 or EcoR1 or PVU2 or BAMH1. What do you think is the answer? If at all we just go behind and see in ROP site, what do you call the protein that is needed for replication, you see the site for PVU2. PVU1 was here. So 1 is always over here, 2 is here, and 3 is on the opposite side. And here you have 2 more. Okay, so always remember that. So it is going to be PVU2. Fine. Now, an antibiotic resistant gene in a vector usually helps in the selection of, if at all an antibiotic resistant gene is there in the vector, that is a plasmid, how does it help us? It helps in the selection of what? Non-recombinant cells, competent cells, non-competent cells, transformed cells. When antibiotic resistant gene was there, first it was there, my dear children, when it was there over here, where is it? I removed that. It's okay. So basically this one was the transformant and the other one was non-transformant. So basically the first thing that you are able to identify is whether it is transformant or not, or transformed cell or not. So this is the answer. Okay? Yes. So other day while I was talking to my colleagues, okay, it came to my understanding that I use two terms a lot, not just two terms. So there are certain things that I repeat a lot, if at all, if at all, if at all, and the other one is basically. And now when I'm saying that, it's all coming in loop in my head. Okay, anyways. Now, my dear children, listen to me carefully for the next thing that we have, and that is separation of DNA. So we studied about vector, we studied about restriction enzyme, we studied everything which was needed until now. But you need to know one more very important thing. When you cut things with the restriction enzymes, what is going to happen? Basically, the DNA is going to be cleaved. The DNA is going to be in parts. Now, you want the one which is shorter, okay, you want your gene of interest as well, and which is the shortest in length. What do you do to segregate them? You do the process of gel electrophoresis. What do? You do the process of gel electrophoresis. So DNA fragments are produced by restriction endonucleases, you got it. Separation technique, gel electrophoresis. Here, DNA is negatively charged and moved towards the anode. So there's a very clear funda. This is your chamber where electrophoresis is going to happen. These are the wells where you are going to put the DNA which you want to separate. In this side of the well, you have positive charge. Basically, you say here you have cathode. So cathode is the side where you have the positive charge. On the other side, my dear children, you have the negative charge. Sorry. It's a slight confusion. Cathode is the one that is negative, and here you have the positive side which is known as anode. So anions are negative, but anode is positive. A for addition, you can remember like that, A for addition. Okay, as of now over here in chemistry you please learn anions, and anions are negative, that is different. So this is anode, and anode is what? Positive in nature. You put your DNA over here. DNA, my dear children, is what? Negatively charged, right? DNA is negatively charged. It is acidic, which is negatively charged. Why? Because it has phosphate group. Now, negative will what happen? Repel, which is why the DNA will start moving from this side and will start going to the other side which is there. Now, the shorter segment of the DNA which is there, it will be able to move the fastest. It will be able to move in the fastest way. And always remember over here, it's not moving in the air. It is basically having a gel over here. There is a gel over here which you call as agarose gel. You get it from seaweed and things like that. And the DNA which is there, it moves within the gel. So basically there is a sieving effect. What effect is seen over here? There is an effect that is known as sieving effect. So it sieves through the gel out there. It's not on the surface. This is a very important question that is usually being asked. So DNA is negatively charged, moves towards the anode. Under what? Under the electric field out there. Agarose gel from seaweeds acts as a sieving matrix. It goes through, okay. Smaller fragments move farther than the larger ones. If at all you compare between larger and smaller one, the smaller one is going to move faster. Now there's a question. There's a usual dialogue that is being written over there. That is, smaller one separates first. Is it like that? No. It can be larger one that can separate first, or it can be smaller one. But who is moving the farthest? It is going to be the smaller one. Anyone can start up the first step. You get it? So if at all in the option it comes that smaller one, moves first, that is wrong. Smaller one moves farthest, that is correct. So we do not know who is going first. It can be larger one, it can be smaller ones. But the farthest, that is definitely going to be the smaller one. Now, basically the DNA fragments which are there, it has separated. Now, how do you view it? You need to view it also. So you are going to view it with the help of one very important stain which is known as ethidium bromide. Ethidium bromide, BR, you can say like that, okay, that's a shortcut. So DNA is stained with ethidium bromide, and now after ethidium bromide, after putting it with staining it with the ethidium bromide also, you will not be able to see it until and unless you are exposing it to UV light, because under UV light, my dear children, it will be giving you bright orange bands. This is important. What bands? Bright orange bands will be visible. That is exactly being shown up over here. Okay, and then only you will be able to identify. Now, basically you have identified the DNA has been separated and you are clearly able to see the smaller fragments are over here. Now, how do you remove this particular DNA segment from here and use it for your own purpose? There's a name of this particular process, and that particular process is known as elution. It's not ILLUSION, it is ELUTION. This is known as elution. What is going to happen? The process of extracting the DNA from the gel is known as elution. The purified DNA fragments are used in recombinant DNA construction. The purified DNA fragments that you get over here, my dear children, these fragments, basically you are going to use it in the, basically again I told you, okay, you are going to use it inside what? The recombinant DNA technology. You take this, put in the vector, and then you do whatever you want. Got it? This is very, very important. Please remember what all important things are there. DNA basically shows a sieving effect only. Small ones does not move, smaller, larger ones, we do not know who moves the first, but the farthest is all obviously the smaller one only. It moves towards anode. Anode is the positive end over here under the effect of the electric field. You cannot view the DNA until unless you put ethidium bromide, okay? Not ethanol, not anything else, it is just ethidium bromide. Now, you also need UV light, because under UV light it will give you what? A bright orange color. It will give you a bright orange color. That is how it usually goes, and later you have to extract it from this particular thing, and that process is known as elution. That is known as elution. Got it? That's all. Cool? Yes. Now, going ahead, you know the basic steps now, right? Now we'll go step by step. You know, in the first part of this particular chapter, I have put so many steps. And I have explained what all you need in these steps out there. Okay, what are the needful? We have explained the basic things. Now, we'll just go step by step and see, are we missing out on anything and things like that. So, the first step that we have is isolation of the genetic material. So whenever we said that we needed DNA for putting inside the host and things like that, you need to understand that this DNA has to be taken out first. How do you take out the DNA? It can be from a different cells. For example, I can take it out from a fungus, it can be from a bacteria, it can be from an animal cell, or it can be from a plant cell. Depending on from where you're going to take it out, you need different things. And the first thing that you're going to need over here is going to be what? Enzymes that are going to break the cell wall, if at all cell wall is included. So there are multiple organisms where you have a cell wall, such as fungus. Fungus has a cell wall which is made up of chitin. And now if you want to break open the cell wall, what will you need? You will need an enzyme that is known as chitinase. Chitinase, okay. Second one, you have plants. And plant cell wall, my dear children, is made up of what? Cellulose. So for breaking open cellulose, what do you need? You need the enzyme cellulase. Okay, that is the second thing. What do you need it now? You need the third thing, that is what? Bacteria. So bacteria, my dear children, they also have cell wall? Yes. What is the cell wall made up of? Peptidoglycan. Peptidoglycan. What are the things that you need for breaking it down? Lysozyme. So remember, in our tears and our saliva we have lysozyme, and these lysozyme are basically there for killing the bacterial cell out there by breaking down the cell wall and everything. Okay, so first thing, we have to get inside the nucleus, but before that, if at all there is a cell wall, we need to break open that. So in fungus you need chitinase, in plants you need cellulase, in bacteria you need lysozyme. Please know this is super important. Now once you come inside the cell, now from this point onwards for animal also it becomes common. Until now animal was not there because animals do not have cell wall. Now when you talk about the nucleus, inside the nucleus, my dear children, you have two things. One is RNA, one is protein. And apart from it, the thing that we need is what? The thing that we need over here is the DNA. So that means we do not need these two guys, right? Right. So if at all you want to cleave the RNA, what do you have? You have RNase. You have RNase that is there for cleaving the RNA, because we do not need that. What about proteins? If at all you want to cleave them, you have protease. You have protease to cleave them. Okay, what about the DNA which is there? DNA, do you want to cleave? No. Which is why you do not need any enzyme for cleaving the DNA. Just remember, RNAs and protease are required right over here. Apart from that, you need all of these things. So first thing is isolation of the genetic material, and this is how you do it. DNA genetic material must be in pure form, free from the RNA, the proteins, the polysaccharide as well as lipids for restriction enzyme action to happen. First thing, cell lysis, breaking open the cell. Use enzymes to break the cell walls, lysozyme in bacteria, cellulase in plant cell, chitinase in fungi. Removal of macromolecules like these things. RNA removed using ribonuclease, that is RNase, proteins removed using protease, and other molecules removed by appropriate treatment. Lipase for fat and things like that. Now DNA is left behind. Okay, DNA is left behind, but this DNA has to be separated from everything else. For that, what are you going to do? You are going to add chilled ethanol. Remember, this is a very chilling experience. So when you add chilled ethanol, the DNA is going to precipitate down, my dear children. It looks like this. Okay, it will not precipitate down. Basically, it is going to separate. Okay, so DNA precipitates as fine threads in the suspension like this. When you add what? Chilled ethanol. So it is not ethidium bromide over here. Ethidium bromide is a stain. This is chilled ethanol that you put. So when it gets chilled, it is going to separate out. You can just remember like that. Okay. So isolation of genetic material done? Done, ma'am. Okay. After isolation, what do you do? Restriction enzymes you use for cutting. Okay, for cutting you use restriction enzymes. So purified DNA plus restriction enzymes incubated under optimal conditions. You cut it. After cutting, what do you do? Basically, you want to separate the DNA according to the length, right? So use gel electrophoresis, about which we have already studied. So first step is isolation, second is cutting, the third one is separation of the DNA in the, in the gel electrophoresis chamber. Now, whatever gene of interest you have got, you have to make sure that you amplify it, because this gene of interest, suppose I put it in the plasmid, it didn't work. So I'll need multiple copies of it, which is why I need to amplify the DNA. And how do I do that? With the help of one very important thing which is known as PCR, polymerase chain reaction. Polymerase chain reaction. And over here, what are things that you require? You need a template DNA, a DNA that is your gene of interest, my dear children. You need 2 primers, and these 2 primers should be oligonucleotides. Okay, short oligonucleotides. Oligonucleotides means multiple nucleotides out there, and always remember, this is not an RNA primer. Here, usually in the case of replication and all, you use RNA primer. Why do you use primer, by the way? For DNA polymerase to extend, to make up a new strand, you initially need something, and that is a primer. Okay, in the case of a normal cell, it is RNA primer which is being used, but here you do not use any RNA primers. This is DNA primers only. Okay, always remember, this is DNA primer. Deoxyribonucleotide triphosphate, that is the thing that you need over here. Now, DNA polymerase, you require the enzyme that is going to make. Now, apart from that, deoxynucleotides, the building block of the DNA. Those all things that you require over here, you also call them as DNTPs. Okay, DNTPs. Anyways, now, listen. There are 3 steps involved over here. First one is denaturation. You have a normal strand. You want to replicate this strand, that means that you have to separate it. How do you do that? With the help of denaturation. Denaturation happens at the highest temperature, that is 95 degrees Celsius. Are you crazy? Just think, 95 degrees Celsius. Okay, it is used for separating the DNA. Now what do? You need to put the primers, attach the primers at both the ends. At what end do you attach it? You need to know that DNA polymerase works in which direction, my dear children? 5' to 3'. It makes a new strand in 5' to 3' direction. So basically in the opposite place it should be attached. So only at the 3' end it will be attaching. Only at the 3' end this particular primer is going to be attached, because in this direction the new DNA is going to be formed. In 5' to 3' direction the new DNA forms. So basically the other strand which is there, it should be in 3' to 5', opposite direction. Got it? So only at the 3' end, this is a picture format question, that is something that you get, and you will be asked which one is correct. So you should know at which position is the primer being attached. So everyone who knows DNA replication knows that it will be only in the 3' end that will come and attach. This is known as annealing. For it, the temperature goes down a bit, it is 65 degrees Celsius. Okay. Now extension. Extension is basically with the help of DNA polymerase, you are going to extend the strand such that you get 2 new DNA molecules. Okay, 2 new DNA molecules out there, that is known as extension. Again over here, the temperature has risen to 72 degrees Celsius. So it was 95, the highest up there, then 65, then it is 72 right there. Okay, and when I say that you require DNA polymerase, it's not a normal DNA polymerase that you take the help of over here. This DNA polymerase is extracted from a bacteria which stays at very high temperature, because only those bacteria will be having an enzyme that can be thermostable. Thermostable means stable at higher temperature also, and you need that over here. All of this is happening at a very high temperature, right? So basically, where do you get this from? You get it from Thermus aquaticus, which is a thermostable bacteria, from where you get what you call Taq polymerase. So first letter from here T and AQ from the second word. So Taq polymerase is the what you call modified or the DNA polymerase that you have right over here. Okay, Taq polymerase is nothing but the DNA polymerase that you have, which is responsible for synthesizing a new strand. Now what do? The cycle repeats multiple times, amplifying the target DNA up to what? Billions of copies out there. There are multiple copies that are being formed over here. And also remember, the amplified DNA can be ligated into the vector for cloning. Now the amplified DNA which is there can be put inside the vector for the next processes to happen. Okay, so please remember all this thing. You need DNA polymerase, you also need deoxynucleotides, otherwise the whole process is not going to happen. You need a primer. The primer is made up of DNA nucleotides only, oligonucleotides, okay, multiple nucleotides. That's all. And Taq polymerase. Works? Direction, please know. Okay, and steps also: denaturation first, annealing second, and E. So D and E. So instead of D and A, it is D and E. You can remember like that. Now, after amplification, what do? You got the multiple copies. You're going to basically put it inside your plasmid, and then transfer of recombinant DNA into the host cell after putting inside the plasmid. So there's one step that is being missed, and that is what? Transfer into the vector. Transfer into the vector, and now the vector that has the recombinant DNA, you are going to put it inside the host cell. You are going to put it inside the host cell. Now in the host cell, it is finally going to express, that is the host cell, it will make the protein. Express means obviously it will make the protein. So after cloning, the foreign gene must be expressed to produce the desired protein which is there. And my dear children, usually it is done in a very large scale production. Now it ends the chapter, do not be worried. So there are two ways you can do it. One is continuous culture system, where what is going to happen is that you're going to give the fresh medium and you're going to remove the used medium. Okay, it keeps the cells in active exponential growth phase. Okay, active exponential growth phase leading to what? Higher yields. This can be done. Otherwise, you have one more thing, bioreactors for large scale production. So it processes large volume, that is up to 100 to 1000. 100 to 1000. And now NCERT's questions are coming like this only. 10 to 100 and things like that. You need to know the small details. So please remember, 100 to 1000 liters of things can be produced over here. It converts the raw materials into what? Proteins, enzymes, and other products out there. You just need to provide the optimal temperature, optimal conditions out there. So this one that you see over here is a stirred tank bioreactor. Apart from it, you have one more, and that is sparged tank bioreactor. So that looks different, this looks different. So this picture is important. You might be asked to draw it up and also label this. So please know how it works. There's an acid base for pH control. So just to understand what is the pH over here, you have a system like that. There is a steam for there's a window for where you pass the steam, and this steam sterilizes everything once it is being done. That is the window for that. And now you have a motor. This motor is required for what you call making this work. This thing that you see is what? Impeller, or basically I can call this as a stirrer. It will be more correct if I call this as a stirrer. Now the stirrer which is there, it has multiple parts. One is the flat bladed impeller. So this thing that you see, over here, they are known as what? The flat bladed impeller out there. And over here you have a broth, the culture broth, where you have the medium as well as the bacteria that you are growing, or the organism that you are growing. And there's a foam breaker at the top. This does not allow the foam to be formed over here. If the foam is formed, it is going to be causing a huge lot of trouble. That is why you have a foam breaker. Okay, and please remember, here you have the sterile entry for the sterile air as well. So air is going to enter through this, and basically when you are putting it here, that means that the stirrer which is there, or basically the impeller which is there, it is going to be responsible for providing the air throughout, right? Now this is a question that came in preparatory, and all of us were like blown up with this, because usually in the picture they have written flat bladed impeller and things like that, and we do not use the term stirrer. But in the NCERT there's one line given, the stirrer facilitates even mixing and oxygen availability throughout the bioreactor. So the question was, which one helps in the mixing of even mixing of oxygen? Stirrer, impeller and things like that. So please know, you just write it as stirrer. Impeller is a part of the stirrer, we can assume like that. Okay, impeller which is there, it is the part of the stirrer. So if at all a question comes like that, it is stirrer. That is your answer. Okay, so there is a cylindrical shape over here with a curved base. Why? It is for better mixing, and there is an agitator system for even distribution out there, oxygen delivery system, stirring or bubbling, and there is foam, temperature and pH control system, and sampling ports are also there, my dear children, which helps us to take some sample and check what is happening up there. Okay, so yes, that can also happen. So either bioreactors or through other medium we can do it. Bioreactors, it processes large volume up to 100 to 1000. And please remember this picture, this is super important. The stirrer is the one that facilitates even oxygen mixing, and yeah, that's all. Cool? Yes. Now after we do all of this thing, there might be things that we require, there might be things that we do not require, and that comes under downstream processing and quality checking. So downstream processing is basically divided into two things. One is separation and the other one is purification. Separation of the thing that we need, that is the protein that we need, from anything else that is not required. Purification is also, after you get it, you need to purify. If at all there are some problems out there, you need to do that. Okay, so separation and purification are the two major goals of downstream processing. So isolate the desired product by removing the unwanted components. But you need to understand that even though we say that it is broadly classified into these two terms, but there are some more things that comes under downstream processing. One is formulation, where basically you are going to add what? Preservatives, to ensure that the product is what? Stable. You can keep it for a longer time. Then, my dear children, there is some quality checking, that is clinical trials, for example, essential for drugs to check safety as well as effectiveness. Okay, and the rigorous checking is being done so that it is understood that okay, it has met the basic criteria out there, and probably we can release it now. So yes, all of it is definitely product specific, but please know downstream processing involves separation as well as purification, these two things. SP, okay, SP, Superintendent of Police, you can remember like that. Apart from that, yes, there is formulation where you add preservatives, and there is quality checking and everything that comes under this. And with that, we have completed the chapter Biotechnology Principles and Processes. So I think this is something that I could do from my end, and I hope you have understood it. Now at this point, everything, if at all you feel that everything is like this, just go and start reading from this particular point. Okay, from this particular point where we had steps involved in recombinant DNA, and you might be understanding things a lot better. But if you feel you have any doubts and you need to ask me, please put it down in the comment section, my dear children. I am here for you guys itself. And if you feel that this session had helped you in some ways, do let me know about that also. And do not forget to hit the subscribe button if you are new to this particular channel. And we are coming with a lot of very important things for your second PUC exam itself. So stay tuned for that also. Lots of love from JJ ma'am. Please take care. Bye-bye.